welcome to the UPLB Museum of Natural History's Biodiversity Seminar Series. I am Julian Afable, the museum's Extension Associate and your webinar host. At this point, I am glad to introduce our speaker today, Janine Kaysee R. Soriano. Kaysee is currently a teaching fellow at the Department of Forest Biological Sciences, College of Forestry and Natural Resources here at UPLB. She is also currently taking her PhD in forestry, specializing in forest pathology. She first encountered fungi when she joined a short biodiversity course conducted by the UPLB Museum of Natural History around the year 2013. Since then, she has been interested in fungal diversity. Her recent works focused on the forest fungi in watersheds and mountain ecosystems in the Philippines. So everyone, let us welcome Ms. Janine Kaysee Soriano.
Good afternoon, everyone. So it's my honor to be part of this webinar, and I'm very thankful for the opportunity given to me by the Museum of Natural History, to Dr. Marian De Leon, Dr. Jen Yim, and all the other staff. Thank you very much for having me here. And for also our guests and some colleagues, students, thank you for attending. So for today, I will share with you a topic about fungi, which is more on the ectomycorrhizas. So let me share my screen. Ayan. So for today, here is the title of the webinar: Fungi with Benefits, Diversity and Conservation of Ectomycorrhizas for Healthy Forest Ecosystems. Okay, during the introduction, it was mentioned that the first time that I had exposure with fungi was during my undergraduate years. And this particular organisms is one of the interesting things that I've encountered so far. So whenever I visit the forest and just walk around Mount Makiling, so there are a lot to see. And for today, I will share with you some of the species that I've encountered during our research trips, and also in different parts of the country. Okay, so first, let's have a quick recap of the mega diversity that we have in our country. So as we all know, the Philippines is one of the frontiers of biodiversity. So why? Because we have a lot of species. Okay, so some statistics say that we have thousands of flora species, some of which are only found in our country. And of course, when we talk about endemism in different regions or islands, so there are a lot to talk about. So wildlife, of course, we have some species that are just found in our country. Invertebrates, so this includes the insects. Okay, so there are really a lot that we can find in literatures. But some of the species that are less given attention are the fungi, and some of the associated ones, the cryptograms. So, yeah. And even though our country is a mega diverse one, so ours is also a biodiversity hotspot. Okay, according to the Convention on Biodiversity, so our country has almost 700 threatened species, thus making it one of the top global conservation areas needing priority. So we've already lost about 2.1% of forest cover annually, which represents, sadly, the second fastest rate of deforestation in Southeast Asia, next to Myanmar. So this one is according to CBD. And why is that so? So our country since we have the chance to document the forest cover, so our forest cover is on a decreasing trend. So based on the forestry statistics in 2015, so this is the current estimate of our forest cover. So we have only 29% of the closed forest, so meaning those that are being will have lesser disturbance. Okay, and while 67% or majority are the open forest, meaning there's already a lot of vegetation that has lost, while the remaining ones are the mangrove forest, or only 4%. And from here, this open, the closed type forests, so our diversity are, say, in Tagalog, nagtitiis in this type of ecosystem, sadly. Okay. And also one of the organisms that face such adversity is, of course, the fungi. Okay, so here are some pictures of some forest ecosystems, okay, that faces such problems. So here in the left is actually Mount Banahaw. And along the foothills is actually a plantation of a vegetable. Okay, during one of our visits around 2018, I guess. So just imagine, we just came over a trail, and then we found this large tract of land having these crops. Okay, so what more if we go to the other parts of the mountain, or even other parts of the Philippines that are rarely visited by people? So we know that there are a lot of diversity that are being affected. So also in the left. So this is actually a part of also in Laguna, in Quezon. And it's one of the areas that I visited for my master's thesis. And the original vegetation here is actually those of the tropical lowland forests. So some are dipterocarps, others some native species, syzygium, like that. And when I talk to one of the guides, so I asked them, why is that this particular tract of land is not that recovering quickly? And he said to me that this used to be a reforestation area in the past. However, due to anthropogenic disturbances, many people are living there, those that are informal settlers. So they put their pasture, their animals there, and some of the seedlings are being grazed upon. So sadly, so that's one of the perspective that we miss when we look at conservation. So the human aspect. Okay, and in line with this status of the forest ecosystem, so what makes fungi or fungal documentation in the country quite a challenge is that they are very reliant on this habitat, on the vegetation. So first, let's have a recap. So what are the fungi? So I guess to those who have attended the previous webinar about mycology, so it's already discussed. But let me just give you an overview of this organism. So they are actually very diverse and ubiquitous. So diverse meaning all over the world, there are actually an estimated of 1.5 million species from Hawksworth, while some other estimates say that no, there are actually 2 million or 4 million. So it varies. But the idea here is that they are very diverse. Okay, we don't have the lifetime to really document all millions of these species. So another is ubiquitous, meaning they can be found almost everywhere. So the very simplest fungi are those that can be found in the kitchen or in the refrigerator, okay, the molds, or even the yeast that is being used in bread. But in the forest, so they can be found almost in any organic matter, even in living plants. Okay, so also the fungi are one of the primary decomposers in the tropical forest. So why is that so? These fungi have the ability to degrade compounds or some of the carbohydrates that they consume to grow. Okay, so without the fungi, our forests will have a very thick litter or leaf litter, I mean, or there are a lot of slabs of wood that are that prevents us from visiting the trails or even Mount Makiling. Okay. And aside from being decomposers, so they are actually associated with some plants, meaning there is symbiosis. So when we say symbiosis, one of the organism is being benefited and the other is also benefited. So that's how unique these organisms are. And apart from that, there are also some that can be used for that have antagonistic properties, that such we can use them for biological control. Others, they can be parasites to control a specific population of invasive insects. But some are also endophytic, meaning they do not really cause harm or good to the plants. So it's just they're just there doing their thing or just trying to live without affecting anybody. Okay. And what's more important when we talk about the forest is that these fungi actually are indicators of forest health and soil fertility. Okay, so various literatures say that the more pristine is the condition of the forest, the more vegetation there are, the less disturbed, we can see more diversity of the fungi. And in return, so their usefulness in ecology, meaning for instance, fertility. So this fungi has such very big role in maintaining the health of the forest. As I've said before, so plant and fungi have diverse relationships. And here are some pictures showing how or where do fungi thrive. So some of them can be found on the leaves, so they can be saprobes, pathogens. Some are decomposers of wood, and some are food to invertebrates, some insects. Okay. But one of the amazing relationships that fungi do is the symbiosis. Okay. So mycorrhizas actually came from the word myces, meaning fungi, and plant roots, meaning rhiza. And when we talk about mycorrhizas, the relationship is mutual. So why? In simpler terms, what happens between the fungi and the plants? So, some part or some portion of their organs, for instance, the hyphae, the mycelium, so they come in contact with the plant roots. So it's like there is a marriage, there is an intimate connection between these two organisms. And in that way, so a lot of benefits arise. And also there are two types of mycorrhizas. So the first one are actually those that are found underground or in the root, in the soil near the root area of the tree or of the plants. Okay, so these are called the AMF, the VAM, or the endomycorrhiza. So they are very microscopic, so less than 1 millimeters, so around 40 to 800 nm. And they look like this, very round, colorful spores that can be found under the microscope. And another type is the ectomycorrhizas or the ECM. So in contrast, these fungi are macroscopic, meaning they have big fruiting bodies. We can see them using our naked eye. So, so what happens is that the mycelium from the fungi unites with the roots to form a structure, and through that structure, the plant can acquire minerals farther than what the roots can find. So that's why there is enhanced nutrient acquisition. Okay, so I hope I was able to explain this kind of relationship in a simple way. Okay, so let's go to the benefits. Okay, so what are the benefits of this relationship? So first, as I've said before, it improves the nutrient accessibility and availability supply to plants. Various studies also show that root colonization provides protection to disease-causing organisms. So at times we can observe that our plants may not grow really well. And then we can, when we excavated the roots, so sometimes there are insects or some nematodes that cause the stuntedness or some disease symptoms of the plant. So if we use mycorrhizas in our plants, so we can observe improvement with regards to growth. And that's because of the root colonization of the mycorrhizas. So another is the changes in the morphology. For instance, in root architecture, ectomycorrhizas specifically, they form the heart-ignet structure. Okay, so inside the root cell, so there's a differentiation of a special type of cell. And because of that, this disease-causing organisms cannot enter or penetrate to the plant. At the same time, the root structure also forms knobby or chubby-like structures, which indicates the presence of the mycorrhiza. And also, another benefit is the improved seedling establishment. When we say seedling establishment, it's how the seedling can adapt to the area that you will be planting it and how it can grow even if minimal interventions are being done. And in turn, so there is improved yield. And when the seedling grow better, so there's higher chances of reproductive success or the capability of the seedling to produce fruit, produce propagules, which is one of the very important materials that we need when it comes to reforestation activities. Okay, so another is that with these benefits that plants can have. So imagine if the individual plants in the forest have this kind of association, ectomycorrhizas, including the endomycorrhizas or the microscopic ones that I presented earlier. So we know that our forest can sustain itself. That's why I've titled this slide as fungi are one of the BFFs of the forest, beneficial for forests forever. So important roles in nutrient cycling. So there's, apart from the intimate association, so there's also an exchange of nutrients. The fungi get the carbohydrates it need from the plant. Then these fungi also supply additional nutrients to the plant. So mostly it's phosphorus. Okay, so one of the macronutrients. So another, fungi serves as food sources for habitat and for the other microfauna. There are some beetles that are foraging on mushrooms or even on conchs or the brackets that live on tree trunks. Another is the alteration of the soil structure, improvement of the organic matter content, and the carbon storage. So when we say alteration in the soil structure, so these organisms help in improving the aeration of the soil. So, and in that way, the roots of the plant can further penetrate into the deeper or extend itself to be within the rhizosphere. Okay, so some of the studies show that per hectare, we can actually harvest about 700 to 900 kilograms of the biomass because of the association of the ectomycorrhizal. So just imagine how much is that 700 to 900 kilograms. So it's about 28 to 36 sacks of rice. So it's very many. And also, imagine a tree, a very big tree. So about 20 to 40% of the root biomass is contributed by the ECM fungi. So that's assuming that such tree have already formed this mycorrhiza since it's on its very young age. So that's from the Global Soil Biodiversity Atlas by Orgezi. So we have talked about the benefits to plants, benefits to forests. What about its benefits to man? So some species are good for consumption, but of course, we need to consult some people who are experts in foraging fungi. Okay, but based on a study, so here are some of the fungi that can be used for food. So bolets, truffles, chanterelles, xylotus, actually, and tricoloma. So I think you are familiar with matsutake mushroom. So that's actually a tricoloma species. And in Japan, there's an article which says that a kilogram can cost over $500. Okay, so tricoloma is a ectomycorrhizal fungi, a type of mushroom. And literatures further say that fungi can or contains bioactive compounds with medicinal potential. So polysaccharides that can be extracted from specific mushrooms species have antioxidant, anti-tumor properties, and we can use those to produce medicine or supplements. And how do we know or how can we find these ectomycorrhizas? So the fruiting bodies can be in the form of a mushroom, bracket fungi, cup-like or coral-like structures. But for ectomycorrhizas, most of the common ones are in mushroom form. There are also coral-like structures, or even those that are that just looks like a round ball. So in the forest, we can see them grow on anything, on the soil, leaf litter, living trees, woody branches, bark, and rotting fruits. And most of the macroscopic ones that have ectomycorrhizal associations are from the groups of Basidiomycetes, the Basidiomycota. And when we go to the field, and for instance, we just want to do mushroom hunting. We want to look at the ectomycorrhizas. So here's one or some of the paraphernalia or tools that we need. So for the amateur ones, very important is to take pictures of the fruiting bodies. So in that way, we can see the preserved state of the fungi in the habitat. And at the same time, some tools for collection. Okay. So in ectomycorrhiza, since the fungi are almost found on the soil and they are almost associated with plant roots, so we want to dig the mushroom. Are there mycelium that almost get in touch with the roots? Okay. And we can also get root samples. So root samples, if there are actually hyphae penetrating the cells of the host plant. And we'll do a microscopic examination, add some stains, and examine it. So it's one of the basic way we do fungal assessment. So now let's look at the diversity. So since we know that ectomycorrhizas are also the macrofungi, so I look into some of the literatures if there are really ectomycorrhizas that can be found in our country. So I just look at the studies from 2001 up to the first quarter of 2022, I think. Okay? And based from this information, so we see that every year or almost every year, there are actually publications that talks about fungi. And we can also see the trend. So it's increasing. So previously, when I first do my thesis in fungal diversity, so there are very little, or it's almost the same authors that I'm looking at the internet. But now there's already a lot of interest. There's a lot of younger individuals who do fungal diversity research, which is good because we can get to see more of the ectomycorrhizas. So we have these statistics showing that there are actually 4,698 species of fungi in our country and 1,031 genera. So this one is from Kimio in 2002. And just recently, there's a publication which say that there are actually 376 validated species names of the macrofungi from 66 families and 130 genera. So take note, these are those that has only species names. So I believe we can get more estimate if all of the reported species in the literature were actually validated up to the species name. And more. Yeah. So we hope to see more of the fungal diversity in the coming years. So in 2015, so there's actually 136 species reported. 2020, 118. And in 2021, with 477 species reported. So this came from 3 publications in 2021. So one of these were authored by us. Okay, so looking at this literature, where are the ectomycorrhizas or where are the macrofungi? So here is a heat map which shows the diversity. Okay, so higher means the color is almost whitish, and the lowest have a bluish or purplish tinge. Okay, I'm not sure if I can zoom this. Ayan. So most of the studies are concentrated in this region. So around region 4, some here in region 3, region 2, and region 5 is already starting documenting their macrofungal diversity. And here are the number of species reported so far. So how much are found in region 4? So there's a lot, of course, because we have Mount Makiling, so and a lot of protected areas all over this region. And I think last year there's a publication which described the diversity of fungi in Palawan. So they also get a very high number of species. So much of these numbers were contributed by one of the previous speakers. So that's Dr. Tadiosa. And we hope to see more of the fungal diversity in the different parts of the country. So in Northern Luzon, in Visayas, and in Mindanao. So I'm sorry, the picture was cropped. But anyway, so upon release of this book, we hope to see more of the diversity. We can see how much or was already covered by fungal expeditions. So in this book also, you will be able to see the fungi in Mindanao and Visayas. So, and those that are not yet really published in literature. So I hope that the viewers will support this book. Okay. And going back to our keyword search of the macrofungi species, so how many are the mycorrhizal ones? So how many? So very few studies describe that the species they found are mycorrhizal. The species that they found are saprophytic ones. So we really need to look deeper into the data that they have. So that's what I did. So I checked the publications, counted how many of the families, how many of the genera were considered as mycorrhizal. And here's what the literature say globally. So there are actually 6,000 ectomycorrhizal species, which came from 20 families of Basidiomycota and 7 from the Ascomycota. And key observations about the ectomycorrhizal diversity. So most of them are associated with habitat or vegetation types. So they're, of course, ectomycorrhizas in the temperate regions are not that common, or some are not found in the tropical areas. Although we have some fungi, temperate species fungi, such as the bolets, that can be found in Baguio. So there's a study which described the ectomycorrhizas there. Further, some ECM species are host specific. So such as the pines, pterocarp, some eucalyptus. And from the data that we have in literature, so there are actually 25 families, 34 genera, and 92 species that are already reported in our country, which is just about 2% of the total ectomycorrhizal species. So very, very small. And here are some of the families. Amanita Astraceae, Bolitaceae. So most of them are the bolets. Cantarelliaceae, Coltricaceae, and more. And now let's look at the ectomycorrhizal diversity that we have found in a few of my studies and researches. These fungi that I will show to you were actually found in tropical lowland to submontane forests. So meaning in elevations ranging from 700 to 1,500 meters above sea level. And most of these are associated with trees in natural and agro ecosystems. So agro ecosystems meaning there are actually protected areas that have been converted into plantations for agriculture or some fruit trees. And in these areas, so there's a mean annual precipitate estimate of 1,600 to 4,500 millimeters. And the mean annual temperature estimate is 23 to 30 degrees Celsius. So here we can, if we look at the map, so the fungi that I will share with you were found in these areas. So some are here in region 4. There's Mount Makiling here, Mount Banahaw, protected landscape, and QPL. And some are here in Bombongan area. Okay, so here are some of the ectomycorrhizal mushrooms that I found. So these two here actually came from the same family, Rusuliaceae. And this one is Rusula virescens. This one is Rusula mariae. So we found this in Banahaw. And based from my our observations, we found this fungi in this under this vegetation. So in the pterocarp, Parashurea malaanonan, near Dipterocarpus grandiflora. And we also found it near a palm species, Areca catechu. So although there are some studies, traditional studies say that the ectomycorrhizas are only associated with some of the gymnosperms and angiosperms, but yeah, so maybe there's another species there. So aside from palm that's actually connected with the ECM. So this one on the far right is actually Amanita. So what's very visible here is the presence of this ring. So it's like a skirt that's attached to the stalk of the mushroom. So that one is Amanita fuliginea. So host associations, Shorea polysperma, a few of the Anisoptera, and Dipterocarpus grandiflorus. So that's according to Sims in 1997. So this one is Tricoloma flavovirens. So it's found in the mixed forest, mainly intermediate trees with agroforestry crops. So what are some other prominent plants that can possibly be associated with this ECM? The santol plant, cocos, and Dracontomelon edule, Pandanus simplex. Okay. And based from observation, so most of these fungi are found in denser canopy forests. So meaning the vegetation is quite rich in those areas. So maybe they like minimal sunlight, they like the moist condition. So that's why they tend to live there. So another type, the Hygroforaceae. So this one is Hygrosibe. and the hygrophorus. So, Brundret in 2008 say that hygrophorus is probably mycorrhizal, so probably. But actually, I found this species under an agroforestry area, under Lantium domesticum, which also associated with some other plants like cacao and citrus. So this one are actually boletes, so boletus. So I think this one is boletus or Serocomus destitutus. So the host is red lawan in Quezon Protected Landscape. What's the difference between the normal bolete and a mushroom? So mushroom have gills like this one, and boletes have pores. So there are also ectomycorrhizal fungi with coral-like forms. So they are the unconventional ones. So why? They look like corals, but they thrive on the soil. So like this one, Cytonopogon, some clavariales mushrooms, Clavunilopsis, clavaria. And when I extracted one, it's actually 3 inches long and longer. So I found longer fruiting bodies. So where did I found this species? So along agroforestry area, so planted with coconuts, some with fruit trees. And when I also excavated another, this one, so it's actually found near the roots of the host plant. So other ones, the earth balls, so they are actually ectomycorrhizal. So this species, scleroderma, so this one is citrinum. Okay, so at first glance, they look like just seeds of a certain plant. But when we pop the fruiting body, so it has pores inside, and if we dug out the mushroom, we see the extensive mycelium. Okay. So oftentimes these are found associated with hardwoods in dense canopy. So now let's go to the conservation of the fungi. So a while ago, I presented to you that we have an estimate of almost 1 million species of fungi. But based on our literatures, so it's only roughly about 2% are the ectomycorrhizal ones that were found in the Philippines. So which gives us a lot of things to do for those of the viewers who wanted to do research on these particular kinds of organisms. So I also found literature which say that in terms of the conservation status of the fungi, only 56% have known conservation status. So meaning, are they vulnerable? Are they nearing extinction, endangered, threatened? So 56 out of the 2 million. And what about in the Philippines? So that's a big question. How can we conserve these organisms? Even so, there's continual increase in biotechnology research, and the companies, industries are fighting over the patents. So which tells us that these organisms has a lot of potential. So not only the ectomycorrhizal one, so the kingdom of fungi itself. And actually, recently we have, or just we already have progress with regards to production of fungal inoculum in our country. So, and some of these are being sold in the market. And also with regards to conservation, we have this question. So what should we conserve first? Is it the host, the plant itself, or the fungi first? So that's quite a big controversy. But we know that if we care for the host, for the vegetation itself, so we know that the fungi and all other species will be conserved as well. So another problem is the seasonality of fungi for consideration in monitoring. So a lot of the diversity research that I've read before, so the concern is mostly on a single ecosystem, and then the sampling at times it's very limited. So if we want to really conserve a particular fungal species, so we look into the long-term objective, monitoring. When do these fruiting bodies appear? When can they be harvested? Where are they in mycelial forms? So we need to know those information. Also, to harness the potential of species as food, medicine, nutraceuticals, and others. And I believe our government has discovered or has been using mycorrhizas for their reforestation programs, which is a good thing because a lot of interest is being put on this species. So this one is with regards to the monitoring and assessment of the terrestrial ecosystem. So actually we have a handbook on the biodiversity, and here is a flow chart. Okay, so we follow the standard procedures, establishment of the study area, doing the sampling, and evaluating the results. However, with regards to the assessment of the diversity, something is lacking here. It's floral assessment, faunal assessment. So what about the fungi? What about the fungi assessment? So we are very interested to see where are the dipterocarps or where are the rare plant species. But there are also much rarer organisms. So that's why it's we hope that those biodiversity studies would also include these organisms. And conservation also includes strengthening research collaborations, the local community, government, academia, and the private sector. So we had the chance to do some regular assessment of the fungi in Mount Makiling through one of my class. And I'm glad to see that some of the students were actually very enthusiastic about finding out those beautiful fruiting bodies actually exist in the forest. And with that experience, so I realized that we really need to engage more young individuals to really do this kind of research. And another is the upgrading of the culture collections in the mycological herbariums. So in some countries, they are updating their collections online so we can cross-validate what we have found here and see if we have similar characters with the specimens deposited in those areas. So it's also one of the possible ways to know or to conserve the fungi. So another is citizen science for the digital age. So when we talk about citizen science, so it's a way to supply knowledge using social media. So, but those people who are contributing do not have to be really experts in the field, but those even normal people can do that. Just they just have the tools, the camera, or the interest to the species. And I believe that we can populate our databases, our museums with more rare and more important fungal species through the participation of more people. And with that, so to save our forests also means to save important organisms like the fungi. So a lot of us want to populate our forests with trees, but sometimes we forget the organisms associated with it. So it's like going into a marriage without a partner, which is sad because we know that somebody losing another, so is inefficient. So there will be troubles in the long run. So it's the same with conservation. So there has to be a synergism with the components of the ecosystem. And let's define what's a sustainable ecosystem. One that over the normal cycle of disturbance events maintains its characteristic diversity of major functional groups, productivity, and rates of biogeochemical cycling. So it's an ecosystem that can sustain itself without much human intervention. So it's one of the things that we hope to achieve, why we do biodiversity studies, why we want to study more about these organisms, to return the productivity of our forest ecosystems. And I prepared an acronym about ECM. So it's basically about engaging civil society towards conservation and management of forest through the beneficial fungi. So that's what ECM is for. It's an effort not made by one person, but it's something that's being done by the collective society. Okay, and with that, so thank you everyone for your kind attention, and I'll be accepting questions now.
Yes. Thank you, Ms. Casey, for that informative presentation. For our open forum, let me introduce our moderator, Dr. Jennifer Niem, university researcher and currently the curator for macrofungi at the Mycological Herbarium at the UPLB Museum of Natural History.
Okay, thank you very much, Julian. And again, thank you, Professor Kaysee Soriano, for that informative talk. Not so many people study macrofungi, and more so in the Philippines. And so I am glad that through your talk, additional insights on macrofungi were provided to the audience, specifically on ectomycorrhizal fungi. So let me check the chat box if we already have the question. And we do have one. The question is from Cheryl Margaret. So her question is, I would like to ask, how can we determine if the association between fungi and other organisms or microorganisms are species specific?
Okay, so thank you for the question, Miss Cheryl. So how do we know if the fungi and other organisms share a specific relationship? So one way to know that is to actually observe them in nature. Okay, for instance, in mushrooms. So can you see this specific species growing on another species or near another species? So that's one of the simplest way. And another is growing the species in the laboratory or doing culturing isolation procedures, which is quite tedious. And through that, we can establish the association. Can the plant respond to the fungi? Can the plant grow better or can produce more dense roots because of that fungi? Or maybe we don't know that fungi is possibly pathogenic. So that's why we need research. It's actually, you can also look for publications if the species that you have in mind also, maybe may ibang studies na nakapagsagot na. So, ayun. I hope I was able to answer it.
I hope that's clear, Cheryl. So there are two ways. One, you can check in nature. So if there is constant association between your mushroom or macrofungi and the host plant, and also in the lab, if they can provide mutual benefit to each other. So thank you for that question. So we have another question from Herman Pan, and his question is, do we have an open database for ECM root morphotypes? And that's one. And the second question is, in regards to conservation of fungi in ecosystem, how do we take into account the mycelium versus fruiting bodies in regards to species count? First things first. So would you know if there's open database for ECM root morphotypes? I don't think so. At least not in the Philippines.
Yes, what Ma'am Jen said is true. So I've been looking for databases, but most of them are from other universities, so not really in our country. But maybe we can develop one through citizen science. And for the second question, so I'm not sure if I get the question
If I may
right.
mention, nagsisimula na kami ng online database sa Mycological Herbarium. So, hindi pa lang siya published online, but we're already working on it. Pero yung classification, if they're mycorrhizal or kung ano mang function, hindi pa included sa database namin. But that's something to think about na pwedeng i-include. So, thank you. The second question is, how do we take into account the mycelium versus fruiting bodies in regards to species count?
I think his question is how to do the diversity counts, right? So what I'm doing, if just in the mycelial stage, so hindi ko muna kino-consider sa data. But if we have already the fruiting body, so we can already count it, pwedeng individually, pwedeng as a colony. So it's really, it depends on the aim of the researcher, on how specific you are with regards to species count.
There is a clarification on the second question. We usually count fruiting bodies for diversity assessment, but multiple fruiting bodies can arise from a single mycelial mass. Yeah. So we can't really say kung there's this mass of mycelia, we can't really say how many fruiting bodies will arise from that mycelia. Is that correct, Kaysee? Yes.
So it's really quite challenging.
Kasi depende rin yan kung sa germination, ganyan. So pag successful yung germination, then malaki yung chances na more fruiting bodies will arise from the mycelia. So pag less naman ang germination, then there would be less fruiting bodies that would come out from the mycelia. So we have another question from Nieves Capili. ECM are symbiotic with host plants, but can they be cultured independently? I mean, I think her question is, pwede ba siyang i-culture sa lab without the host plant? Would it grow in artificial media?
Based sa mga studies na nabasa ko, laging may host plant dapat when we culture the ectomycorrhizas. Kasi when we say ectomycorrhiza, they are in obligate association. So meaning, pag wala ang isa o wala si plant, so hindi talaga mag-grow or mag-germinate yung ectomycorrhizas. Naka-mute ka po, ma'am.
We have another question from Nieves Capili, Ma'am Nieves Capili, and she asked whether there are specific seasons where the ECM footing bodies are best available for field observation.
Okay. So, in temperate countries, meron silang season na talagang yun yung inaabangan nila to collect the macrofungi. But here in the Philippines, I think one of our basis is yung presence ng moisture. So halimbawa, may okay yung rainfall in that area. So possible na ito na yung time wherein active na yung mycelium. So pwede na siya mag-reproduce ng fruiting bodies. Merong mga records na may specific tawag dito, season or regions or quarter, pero yung months, so I think hindi pa siya talaga na
I guess more on ano siya,
specify.
kapag may rainy season, mas marami
po.
yung ectomycorrhizal
Fruiting
fungi
bodies.
that you expect to grow kasi kailangan ng moisture as Professor Soriano mentioned, kailangan ng moisture for production ng fruiting bodies. For the germination, sporulation, ganyan. So, moisture ang kailangan. So, probably, mas maraming ectomycorrhizal fungi to expect around May to December, siguro, or November, kung kailan maraming ulan. Do you agree, Ma'am Kaysee?
Yes. Actually, yung based dun sa mga pinosend kong pictures, mostly sila July, August. Ayan. Pero we have climate change. So it's really nagva-vary na ngayon yung seasonality ng occurrence ng fruiting bodies.
And then we have another question from Ma'am Eldrin Arguelles. What is the advantage of using ectomycorrhiza as inoculants for enhancement of plant growth as compared to PGPRs, which is also very popular in biostimulant research?
Okay, so thank you for your question. So I'm not really familiar with the PGPRs, but I guess both aims to really improve the growth and the development. So, yeah. And yung sa advantage, so I think it's almost the same. But if we really want to see yung specific details ng mga advantages na yun, so I think we really need to do more research and compare it, lalo na kung we are testing it on a same plant. Halimbawa, we want to use ectomycorrhiza for Pinus cassia, pine. And gusto rin natin makita yung response nito with PGPR. So, we need more information to really know ano ba yung advantages.
Thank you, Ma'am Kaysee. Meron lang akong quick question. I'm just curious to know, are all terrestrial mushrooms that grow in close proximity to a living host plant considered as mycorrhizal fungi or mycorrhizal mushrooms?
Ayan, so it's also one of my assumptions before when I'm just starting out with doing this diversity research. So, hindi. Hindi palagi. Kasi merong mga species na possible pathogenic sila sa roots. No? So, there's, halimbawa, yung mga lepiota. So, we can see them in the soil. They look like ectomycorrhizal mushrooms. Pero yung pala, mga lang sila, decomposers, saprophytes. So, we need to really see yung saan ba nakakonect yung mycelium or kung better, mag-grow natin si mushroom na ito on a nursery or field greenhouse study and really determine the association. So, ayun nga. So, not always magandang or basis yung soil.
Thank you for that answer. And then we have another question from Chabelita Avikivil. We saw that most diversity studies were done in Luzon. I may have missed it earlier, but are there current projects focusing on the EZM fungal diversity in Visayas or Mindanao?
Okay, so thank you for your question. Based sa pinresent ko na map kanina, 'di ba, more on ano to sa Luzon areas. So ito kasi yung na-publish na literature. But actually there are a lot more fungal diversity research na hindi pa napapublish. So that's why di ko maipakita talaga yung map kasi nasa book kasi yon. So you can see na meron na actually sa Visayas, sa Mindanao, even in the previous, yung mga old pa na mga expeditions. So meron nga sa Sulu area. So actually, with regards to publication of the researches, yun lang yung medyo mabagal tayo. That's why it's hard to really have this complete database of the ectomycorrhizas.
The book will be published in October, so it's coming out soon. Intayin na lang nyo. Yes, thank you. And then any more questions? Okay, so I have another question. Can ectomycorrhizal fungi be used in my garden of, for instance, ornamental plants to obtain the benefit that can be derived from the ectomycorrhizal association?
I think that question is also related kanina kay I think si Miss Cheryl yung nagtanong about sa species specificity. So, ang ectomycorrhizas kasi, lalo na yung mga specific na species, I think yung mga boletos, medyo sila, choosy sa host plant. So, hindi lahat talaga ay nakaka-associate nila. But based din sa mga, nakita ko rin sa aking sa field work, no? So, yun yung kulang sa literature. So, hindi talaga nakikita yung mga native species natin that they can actually be host of the ectomycorrhizas. Now, for the garden, I think kung halimbawa, mga picara lang yung mga halaman or yung mga ornamentals lang talaga, we have another mycorrhiza to use for that. And that are the vesicular or vascular mycorrhizas. So, hindi sila yung nagpo-form talaga ng malalaking fruiting bodies. And for sure, mas mataas yung chance na improve niya yung growth ng halaman kasi almost 90% of plants can really associate with the endomycorrhizas. So that's my suggestion if you want to use mycorrhizas for your garden.
Quick follow-up lang. Meron na bang commercially available na ECM sa market?
Ayan. Based sa mga nakita ko pong studies, so I think the biotech has already produced an inoculant, specifically the ectomycorrhiza. So they use Physolito, some Scleroderma species, and they also got it from different plants all over the Philippines. So I'm not sure if still in production pa po ito. So I think matulungan ako ng mga taga-biotech if it's really still available pa. Kasi so far nakikita ko pa lang po ay yung sa Mycobam. Ayan. For the other, halimbawa yung mga private industries, I'm not sure dito sa ating bansa, pero overseas, they really use the ectomycorrhizas for plantation ng mga pines, mga populus, ganun, Pinus sylvestris. So they already have product for that.
Thank you. We have a question from J. Elorde, and the question is, apart from using dichotomy keys in identification, what other tools can he use for identification of macrofungi? Kasi he's asking because his undergraduate thesis is on the general listing of macrofungi in Mindanao, specifically in South Cotabato.
So about identification, so Actually, dichotomosky is one of the best way to really narrow down the identity of the fungi. But yung problem natin in our country is kulang yung dichotomosky, lalo na for species level. So I think other than that is of course consultation with the experts, with those who are really doing the fungal diversity research. And we have the Museum of Natural History and some other scientists in the academy doing that. So yun yung pwede ko pong suggestion sa kanya. Makamute ka
Also,
po,
we
ma'am.
have a number of books na pwede nyo i-consult on fungal identification. One is yung kay David Aurora, kaya lang it's more on mga macrofungi or mushrooms from North America. But at least yung genus level, magagamit naman natin yung book for identification at least sa genus level. Kaya lang for species ngayon, you have to do microscopic work and preferably molecular work. Kaso walang pang masyadong work sa molecular when it comes to identification of macrofungi in the Philippines. But there are a number of references that you can consult. I have another question. Would you know if there are endemic ECM in the Philippines? Kasi meron akong nabasang isang publication from, I think, from Nueva Ecija. Endemic daw sa Philippines yung Colibia reynakiana. But when I check it out sa website, online database sa mushroom, international online database on macrofungi, hindi siya recognized sa Mushroom Observer tsaka sa Index Fungorum. So would you know anything about this?
Actually, it's also one of the things that I'm curious about sa endemism.
Parang walang walang
Wala
report
pa,
at the moment.
Opo.
Oo. Although dati meron sinasabi sila na Termitomyces, species of Termitomyces, but they found out na makikita rin to in other parts of Asia. So at the moment wala yatang endemic endemic fungi, macrofungi in general. So I guess we don't have any more questions. Let me check. Yeah, so it seems like we don't have any more questions. And I guess we can end now our Q&A. Thank you, Ma'am Kaysee, for graciously answering all the questions of our audience. And I will now turn you over to our MC, Miss Julianne.
Now, let me present the Certificate of Appreciation to our speaker. So, the University of the Philippines Los Baños and UPLB Museum of Natural History presents the Certificate of Appreciation to Ms. Janine Kaysee R. Soriano for serving as resource person during the UPLB Museum of Natural History Biodiversity Seminar on Fungi with Benefits, Diversity and Conservation of Ectomycorrhizas for Healthy Forest Ecosystems, held on September 5, 2022, 2:30 to 4:00 p.m. via Zoom, signed by our Museum Director, Ms. Marian P. De Leon. Thank you, ma'am, for giving your talk today. So before I proceed to our closing, let me just share a few reminders about our social media pages that you may follow for further announcements of our future biodiversity seminars. The details are flashed on the screen. We have a website at mnh.uplb.edu.ph and our official email address mnh.uplb@up.edu.ph. We are also on Facebook, Twitter, YouTube, and Instagram at UPLB Museum.